All right, this is unit 4.4, and we're talking about cell division. What we have on the screen is example of mitosis. So to start with, we have the parent cell on the very top, right? The parent cell has two chromosomes in its cell. And after the process of mitosis going through PMAT, and then after cytokinesis, we have two daughter cells with two chromosomes in their cell. And we can see that the chromosome number stays constant. This is because the chromosomes in the parent cell were replicated before the process of mitosis. So the parent cell would look like this before the splitting. And then it goes through the process of mitosis, and we end up with two new cells, daughter cells with the same chromosome number. So this is an example of how chromosome number stays constant in cell division in mitosis. Next, we're going to talk about meiosis. Meiosis is the process of how gametes in our body are made. So, eggs in female and sperm in males. So we're going to learn how male gametes are made in this example. Let's say you start with one chromosome, right? In human, we have 46 chromosomes, 23 pair, but we don't have time to draw it out. I'll just use one chromosome for simplicity. So meiosis occurs in two stages. That means we start with one cell, parent cell, and we end up with four gametes. Okay, so actually, you know what? Let's do two. Okay. So to start, we need to know the difference between diploid and haploid. Diploid just means a pair, and haploid means single. And we usually use these two terms in biology to describe the diploid number and the haploid number. What we see here is a diploid cell because we see a pair of chromosomes, right? And after they each split, remember this is the process of meiosis. So the first splitting would be called meiosis 1. Its process is similar to mitosis, apart from the separating of the chromosome. In meiosis 1, the chromosome pair, what we call the homologous chromosomes, are separated. So these two are homologous pairs, and that one single thing is a chromosome. So the homologous pairs separate, leaving us with one chromosome in each daughter cell. Okay, so we start with diploid, and we end up with haploid, and this is because we don't see a pair of chromosome anymore. There's only one. You see one chromosome, no pair. That's why we call it haploid. And so this is meiosis 1, finished. After a while, this daughter cell then goes to meiosis 2. Meiosis 2 is similar. It goes through prophase, metaphase, anaphase, telophase, and then cytokinesis, right? But the difference is, again, in the separation of the chromosome here. So we would have 1, 2, 1, 2. Actually, like that, 1, 2. So this is this is identical to the splitting of chromosome in mitosis. Instead of having the homologous pair splitting, like in meiosis 1, meiosis 2 splits the sister chromatids. We know that sister chromatids are like one of these, just half, and they are joined in the middle by the centromere. Actually, right? Yeah, you get the point. So we start with diploid up here. We end up with two haploid cells, and out of these two, we again have four haploid cells. And you may ask, why are they haploid? because you don't see a pair. And also, what we need to know is that the sister chromatids are identical, so they contain they contain identical information, which means meiosis one would lose half the DNA mass and half the information, whereas meiosis two doesn't lose any information, but it loses half the mass. You get the point. All right. And since this is haploid, right? This is a male gamete. A male gamete is haploid, which means it has half the DNA information needed for an organism, and a female gamete. When these two fuse together, that's the word. When these two gametes fuse together, that means a haploid gamete joins a haploid gamete. They're going to join to form a diploid cell. And that's how chromosome number is restored in humans. This the process called fertilization. It's beautiful. And that's all we have to learn in 4.4. There you go.